My name is Janet Thornton. I have worked at EMBL-EBI since 2001 when I joined it as a director. I stepped down as the director in 2015 and reverted to being a group leader again, a research group leader, but only for 3 days a week. So I've been part-time for the last 8 years.
What inspired you to work on bioinformatics?
Well,
was
I
that
never
always
really
your field?
intended to work. That was, I trained undergraduate as a physicist, and then I knew I liked the process of physics, but I don't like machines. The thought of going into, I was interested in atomic particles, but I wasn't keen on the idea of synchrotrons, and I wanted to do something more associated to life and medicine. So I did my PhD in biophysics at Mill Hill, National Institute for Medical Research, mainly because I think it had medical in the title. And I did some biophysics there. I also did like a conversion course between physics and biophysics at King's in Drury Lane in London. And that was, for me, was incredibly important because I knew nothing about biology.
I was going to ask if you'd ever studied biology. No.
Not at all. I hadn't done my final years at school. I'd not done biology. I never set out because, frankly, when I was doing my PhD, I hadn't done any programming because there weren't computers.
Yes.
And the first machine I worked on was this sort of desktop calculator, but you could program it. So I started doing this, and it was a turny one, and I mean, it was ancient looking now back at it. But then I learnt to program, and I enjoyed that. And the computer that we had at Mill Hill was rather special because you could look at molecules in 3 dimensions. So we had the mirrors where you have 2 views of the same molecule, and by adjusting the mirrors you could see them in 3 dimensions. And I just enjoyed that. I just thought it was really neat. And so I started programming, and the things I was looking at were actually one of my first tasks was to draw out the 20 amino acids, which stood me in good stead because I actually learnt what these amino acids looked like and what they were, which I had no clue of prior to that. But I was doing circular diachroism, which looks at the conformation, and in particular studying the coenzyme nicotinamide adenine dinucleotide. But as a physicist, I didn't have a clue what it is. I should say I hadn't done chemistry either since I was 14. So my knowledge was pretty minimal. Maybe it still is, I don't know. But anyway, I went on, and of course there wasn't a field of bioinformatics. No. I mean, the name wasn't invented until 91. That's 20 years after I started my PhD. And people, of course, the people in genomics thought that they'd invented computational biology, whereas in the protein structure world, we'd been working with computers because they were necessary to determine the structures. So we developed a lot of computational tools to handle the geometry of proteins, really. And so it wasn't planned. I don't think any of my career has been planned, Angus.
Well, I think that's true for many of us. I mean, I was never particularly keen on the term bioinformatics because I think really what you do encompasses a much broader area. And for me, the term informatics is part of it, but it's not all of it. It's
not the guts of it, in my opinion. So if I give an example, we've been looking at enzymes in huge amounts of detail. But I wanted to look at the reactions, and not having done chemistry, of course, there's a whole field of cheminformatics, but I'd not been part of that. Yes. But we developed lots of tools for handling these huge protein molecules, but in comparison, the tools for handling enzyme reactions were hardly developed. And so also a student came to the lab, postdoc, who was a chemist but also a very good computing person. And he pointed out that, you know, reactions were like equations. You had to balance the left side and the right side and make it all work, and therefore you could compute on them. And so developing the system to capture enzyme mechanisms, which is what we've been doing, using some of these cheminformatics tools, it's capturing life in a computer, basically. And that to me is what I enjoyed, so that we can understand it, so we can model it, so we can use it to predict things. And that is quite different from the term bioinformatics.
Yeah, I agree. Exactly. I think we're...
And so it's computational biology.
Yes.
But to me, it's where much of our biology needs to go, because we need to take these processes, we need to have, I mean, they won't be like Newton's equations, because it's much more stochastic and everything, but we need these ways of modelling the processes of life so that we can predict things, so that we can actually understand them better.
And of course the field of computing itself, separate to biological computing, just the field of computing has exploded in 20 years.
So as a student in Mill Hill, I used to use what we call the NAG libraries, but were all written by physicists for doing energy minimization. So they were just programs that minimized a value. Nobody shared other than those, you know, and that was the code, and you would, you know, type it out to get it on the computer. But I mean, for many years, we didn't share our programs because they wouldn't run on somebody else's machine. Yeah. So, and it was very, very different then. You know, I had to feed it, I used paper tape. I had to feed in the compiler to the computer before I could feed in my program, and then discovered I missed out a comma. So you'd have to go back, generate another paper tape with the comma inserted, and then run through the compilation again. I mean, it was so slow compared with now.
Yes.
It was completely different. But I learnt a lot. I have to say I was probably one of about 3 people in Mill Hill using the computer. It was a very specialised little niche. And of course there weren't any jobs when I left.
There weren't jobs, and I guess most people just didn't foresee the impact they would have on all of our lives.
When something good comes along, people adopt it. Yes. And I feel that the same will be true of some of these medical advances that come from computational biology, that when clinicians realise the power of the computing and the information and the knowledge it can use to make predictions, I think it will just revolutionise medicine.
What inspired you to go into science in the first place? I understand that you were inspired to switch from physics into biophysics and then into biology and computing and biocomputing. But very originally, what grabbed your attention?
I don't know really. I thought I might be a geophysicist because I wanted to do the geography and the physics, and I went for my interview for my undergraduate and studies in physics, and I said, oh well, I thought I'd like to combine, because I like rocks as well, and you know, I used to collect pebbles on the beach. I like the different shapes and colours and everything. And the person interviewing me said, well, Janet, there are no jobs for women in geophysics. It would be a complete waste of time you doing that. And I thought, oh, all right, I'll do straight physics, which is what I did. And then I guess I always thought I would be a teacher. That's what I envisaged myself doing. And then I sort of made a deal with myself. This is going to sound crazy, but I thought, well, if I get a first, then it wouldn't be sensible. I should carry on and do a PhD. And I got my first, and I thought, right, I'll do a PhD, but I want to shift into the more sort of medical life sciences rather than the hard sciences. And so that's what I did. And then actually, when I started my PhD, it was tough. I have to say, changing, I found, because I knew no biology, and I just found it really hard to know what was the point of what I was doing. Mm-hmm. You know, I thought, I didn't even know what this molecule NAD did and why it was important and why I was studying it. I mean, I like the computing side of it, but I really had not a clue. Of course, then I got entwined in proteins and things, and I began to understand. I wrote my thesis, I did a lot of reading, and I thought, this is really quite fun. For biology, the great thing is that every living organism has DNA and proteins. And it's amazing, isn't it? Don't you think it's amazing?
It is.
You know, and if you
really
tell
amazing.
people, oh, the plants have got proteins just like you've got them, and actually they're really rather similar. It is quite amazing how you can develop different organisms and how they evolve, yes. Which took me years to appreciate.
Yes. I think it is a revelation to most people that even the term protein, they think of as a foodstuff, if anything.
Yeah,
if you're not scientific, because you have to understand
protein.
The idea that proteins are the targets of almost every drug you would take, well, and in many cases they comprise about 50% of the drugs that you're given there.
Well, that's right, and they do everything in the body.
Yes.
I mean, I still maintain they are rather more interesting than DNA because they do things. You know, everything that happens in the body really is because of proteins, and the DNA is just the silent messenger. Well,
we're all part of a system. We need each other.
We need both. We obviously need both, yeah. And that's one of the pleasures of getting older in science. You remember what it was like. You remember what you didn't know that
yes.
you do know now.
Yes.
And that, so you see the progression of science. I mean, it's a wonderful career to have for that reason, I think.
It is, and clearly you've seen a progression from the founding of bioinformatics as a field to something we see today, where it's just, you know, beyond what one could reasonably have expected. So yeah. I'm going to put you on the spot. Look 10 years ahead, 20 years ahead. Instead, you can look back 10 or 20 years and see these changes.
No, exactly.
Look forward
and
tell us what you see in your the Dame Janet Thornton crystal ball of bioinformatics.
Well,
what are we going to see in 10 or 20 years?
So let me look back first.
Okay.
And say, what did I know in about 1980? And what could I predict going forward? Mm-hmm. So there are the clear, it's like the Rumsfeld's knowns and unknowns. There are the clear knowns, and with the structures, the protein structures that were being determined, especially when I was in a postdoc in a crystallography lab, etc. And you could we plotted very early on the number of crystal structures per year, and watched the trend. And it was entirely predictable. And I can even remember talking with my colleagues saying, gosh, I wonder how many structures we'll have, 20, 30 years from now. And of course, it's now over 200,000. When I started, there were 20.
Yes.
And seeing this, but that was a predictable thing in many ways, although the things that have allowed it weren't necessarily predictable. And in particular, the ability to sequence DNA that I did not see coming at all. And even when it came, I'm not sure I even appreciated the impact it would have on my science. So that was an unknown that has had a huge impact. And the whole concept of high throughput biology, which obviously needs computational biology by the side of it, but it changes the way that you look. Not always in good ways, sometimes in not so good ways, but it changes the things you can do and the way that you think about problems. So to me, there are some predictable things and there are some unpredictable things. So the predictable things, I think, are we're obviously going to get more and more and more data.
Yeah.
We're obviously going to look in more detail at how these the pleiotropy, how all of the pathways interact with each other and have an impact. I hope, so we're going to, I hope, put these molecular things in the context of the cell and understand much more about how that geometry of the cell, which
as a system
works, and how those cells come together as organs or whatever to create organisms. So we're going to sort of have a much better way of stepping from the molecules and the genomes to what comes out at the end. So, you know, if I give you a genome, is it a dinosaur or is it a chicken? You know, are we going to be able to really predict from the genome what the species is? And the answer is probably yes, but we'll do it, I still think of it as cheating. By comparison, we'll look at the chicken genome and we'll look at the dinosaur genome and we'll say, oh, well, it's obvious it's one of these or one of those. But if you could do it from a fundamental perspective,
from first principles.
First principles, wouldn't that be wonderful to do that? So I think we will get towards doing that. I think we will, I think the understanding how the brain works is still not clear. And that was the other thing. I was offered a PhD in neuro as well as in the molecular, and I chose the molecular because I wasn't entirely sure where the neuro was going. And it's taken time. I mean, I think in retrospect, I actually did the right thing.
I think you
totally
did.
By accident, of course. But you know, the advances in my world have been just phenomenal. So I think the other predictable thing is the progress in medicine. And I think the practice of medicine will be completely changed by computational approaches to medicine. It's taken, I can remember when I joined EBI as a director, I said this is going to make a big difference. And it's slow, because adoption of these things is never straightforward. And people operate in one way that they're used to and they don't want to change. But the proof of the pudding will be in the eating. When there are these tools that are really predictive and diagnostic and prognostic, then the clinicians will adopt them because they see that they are good for the patients. So I think that will happen.
Yes.
Whether it will be soon enough for me, I'm not quite sure, but it will happen in the next 20 years' time. And the genomes, you know, the impact of, well, I don't know, the genome sequencing, it's obviously critical for developmental disorders, but it's still very, very complicated for anything chronic or diseases like that.
I mean, we're seeing playing out in real time this burgeoning growth of neural networks and artificial intelligence and what they suddenly enable. And you in the bioinformatics community have this remarkable volume of data that are trainable, can be used to train. So I imagine a lot of the predictions you're mentioning can be fed and fostered by these technologies coming.
Well, yes, completely. I think that will happen in the, and you know, AI methods, the new ones, are really good at combining disparate data.
Yeah.
And effectively weighting them to get the right answer, which makes them very predictive. They don't always, I mean, some of it depends, but they don't really explain why, you know, how things work. But they are for, I mean, the other thing, they're so important for biology because of evolution. And that because things haven't just been reinvented and reinvented, they've evolved. And that means that, as you know, structure is much better conserved than sequence, etc. And the whole evolutionary perspective on biology, which is almost absent from lots of physics. I mean, it wasn't part of my thinking at all, and it took me years to appreciate evolution rules, okay, and it does in biology, but that fact makes AI approaches incredibly
all the more powerful. Yes.
Yeah. And so bringing the data together is just wonderful. I probably can't not mention AlphaFold and the ability to predict structure.
So perhaps you could just briefly explain what AlphaFold is and what it did to the field for some of the people watching this who are less familiar with it.
So the problem that probably brought me into the area of protein structures was to try to take the linear amino acid sequence and from that predict the three-dimensional structure that that molecule falls into. So it's a very easy problem. It's like an infinitely complicated three-dimensional jigsaw. But it's a problem that actually small children could understand. You know, if you give them a shoelace and say, fold it up into something, they'll come up with their bundle of shoelace and whatnot. So the problem is very easy to state. It proved to be extremely difficult to solve. And in the early days, what I was involved with was almost deconstructing the problem and looking at different aspects in great detail of protein structure, which I found fascinating because it was a cause and effect. If you have this sequence or this pattern of residues, then you will get the helix or the turn or the loop. And so there was a logic to it. It wasn't a random thing, it was logical. And so trying to pull that apart, and we used neural nets back in the 90s for predicting beta turns, you know, and I had a collaborator with a neural net specialist and we did these things. But it soon, you know, we tried so many different things, and I realized that the solution was going to be technical. I didn't know what technical, but I knew because the improvements were very in the quality of the predictions of these structures were very slow for many years. And also it was kind of very competitive.
Really?
And although I am competitive when I'm backed into a corner, but I'm not naturally wanting to go in and fight with somebody. You know, I don't like having disagreements and things. And so I thought, no, I'm going to stop doing that because I've no new ideas. I didn't know what to do to make it work. And so we went and started looking at the relationship between structure and function, which is sort of the next thing on, and trying to look at that. In the meantime, the neural net field and the artificial intelligence field, the deep learning field, improved the way that you abstracted knowledge from data fundamentally. And company DeepMind were able to make really quite significant improvements in their ability to predict this three-dimensional structure from the sequence. And not only that, with the tools they've got, they can predict all the protein structures. So they have generated 200 million structures that we at EMBL-EBI have made a database for, led by Samir Velanka. And that, so for almost any protein that is in UniProt, you can get a predicted 3D structure. Some of them are accurate, some of them aren't so accurate, but in general, they are very good. And this has had a huge impact on the field because people begin to think structurally. And for a long time, I think the structural biologists, of course, loved their structures and, you know, would extol them and they knew every infinite detail about them. But they weren't, it didn't permeate that much into other areas of biology. I think with AlphaFold, people realised that if they had a new sequence, they could go and look at the structure. It might not be perfect, but it would give them a good clue. In fact, they could have done that before, but these tools weren't available and it wasn't so easy. And overnight, it became like the internet. Press a button, get your structure. And that actually, so developing this database at EBI that was based on all the engineering they'd done for the protein data bank, the collection of known structures, just came into its own. because it could deal with, instead of 200,000 experimental structures, 200 million predicted structures. And of course there are large language models, different ways of doing it now where they've got 600 million structures. So, you know, it's changed the way that people think about structural biology in a positive way, I think, actually.
Yeah,
although having the gold standard experimental structures is always going to be, at least for some time to come, critical.
Sure. Yes. Yeah, no, absolutely. And it was very interesting seeing, as a reminder, how I think for decades it was assumed, of course it's non-random how proteins fold, otherwise they wouldn't reproducibly fold into the same structure. Thing. But it was, I think, looking from the outside, felt that this was a top-down problem. It was going to be solved top-down, because if you knew enough chemistry and wrote the right equation, you would be able to put together facts of chemistry and stereochemistry and electrostatics
and do it.
And that would tell you why it folds that way. Whereas the actual way the solution came was from left. I'm pointing to my right hand and saying left field, which but absolutely. It came completely from an unexpected. But that's I think that's something to be celebrated. Keep our minds open.
So do I. I think it's just teaching us more about structures, but you have to realise the limitations of these predictions.
Yes, of course, everything.
You can't.
Yes, it's not an overnight solution to everything, but still. But it's also a very exciting thing that EBI was able to play this fantastic role of bringing that new information in an unprecedentedly short time and making it accessible. That would be great to hear how you see the role of the European Bioinformatics Institute to enable, catalyse advances in that way.
So I should say that the European Bioinformatics Institute, for those who don't know, is part of EMBL, of course. It's based in the UK, but we're all part of the one EMBL structure. And within the Institute, which is now about 800 people, 2/3 of those, well no, 3/4 of those people provide data services for biological information to the world, to the point where our website gets 107 million hits a day. I mean, it's phenomenal, this data. And we have all different sorts of data, heterogeneous data, different organisms, different types of data structures, sequences, transcriptomes, expression data, obviously, and epigenetic data, all sorts of things. But the reason, the Protein Data Bank in Europe, which is part of a worldwide collaboration, which is really important that everybody shares their data, and it doesn't matter which country you're in, you can look at the data and access it. They had developed tools for handling these 200,000 experimental structures. But actually, Samir, who leads the PDBE, realized that predictions were also important. And so he had they'd engineered the system that it would take many more structures before AlphaFold ever came on the scene, with a view to taking predictions of structures. And so when actually we were approached as to whether EBI would create this AlphaFold database with all these structures available, we had all the tools. And so within 3 months, that the team was able to generate this database that meant that anyone, anywhere, could look at any of these 200 million structures and whiz it round on the computer and look to see what it looked like. And so I think that preparedness was important, and it was foresight of what was coming, because many people predicted structures. It's just how good they are, the predictions. And so one of the challenges really is the amount of data but more importantly, the heterogeneous data, that you have different types of data, and each of these types of data really requires its own database. But if we try and understand the system and organism, we've got to combine all this data together in some ways. Yes. And that's where I think the deep learning will help us. Yes. I'm sure it will.
And the whole is greater than the sum of the individual parts.
That's right.
But I mean, it's also a great example of the value of flexibility and the realization that we don't know, as you said, the Rumsfeld, we don't know what we don't know.
Yeah.
And that ability to respond quickly and take advantage of doing it. Do you think that message has really got through to our political masters and funders in Europe?
So frankly, it's always difficult to secure funding for data resources. It doesn't matter where they are. Partly because, I'm not taught at all, but I've worked on, over the last 20 years while I've been at EMBL, I've worked on ageing as well. Mm-hmm. It's much easier to sell ageing research than to sell the idea of a database. But actually, the database understands and underpins our understanding and will underpin our understanding of ageing. As we're already seeing, that understanding is increasing. Not that we're there yet by any means, but it's definitely increasing. So I think for the funders, this funding of infrastructure to support the data in the life sciences is really critical. And EMBL, the wonderful thing about EMBL is, of course, we are funded by 28, 29 member states, and they all put in, according to their own GDP, they will put in the amount that they contribute to the whole. And the whole funds the creation of these data resources. So I wouldn't say it's cheap for them, because data infrastructure is expensive. But they can't do it on their own. We have to do it, not only actually within Europe, but within the world, and share the data across the world. And that, the EMBL model for funding really is a huge advantage, I think, for supporting data. Yes. It's something that, you know, Germany on its own, or the UK on its own, or France on its own, wouldn't want to invest, or if they invested, they wouldn't want to share, because they'd say, we pay for all of this, this isn't for everybody. It just
makes sense to do it collaboratively.
But if you do it collaboratively, then it's much, much better for everybody.
Yeah, I completely agree.
It powers science. I mean, one of the nice things about being director, and of course you get asked to go and talk about the institute, etc., and I've got a wonderful slide that shows in real time all the people using EBI resources from around the world. And I used to think, well, here I am just talking about this, and all these people, while I'm talking, are pulling data out of EBI and using it to really empower what they can discover about life. You know, it's a fantastic feeling, actually.
Yes, I think, Janet, your enthusiasm, persisting enthusiasm for science is extremely apparent, and has been an inspiration for your colleagues and for a generation of scientists. And thank you very much for coming to speak with us.
Oh, well, thank you, Angus. It's been my pleasure.
Thank you.